Hello students, good evening all of you. Welcome to brilliant YouTube live classes. I hope you all have finished your physics, chemistry, mathematics and physical education exams, right? It's very well, I hope it went very well. Next exam is biology. So 16th March, our countdown start. So we have 3 days, 13th whole day, 14th, 15th, 3 days. So in these 3 days, we should cover all the 13 chapters. 13 whole chapters we should cover. It's a big task. Especially we will focus on the high weightage chapters like molecular base of inheritance, principles of inheritance, biotechnology, human reproduction chapters. In this class I am planning to discuss principles of inheritance, molecular base of inheritance, evolution chapter, very important concepts, very important questions. And moreover, all of you, I'm telling you, I'm requesting you all, take NCERT and take one notes and sit with me. Because when I explain this question, we have to do a dihybrid cross, test cross, okay, Mendelian cross, mutation problem, especially X-linked chromosome problem, X-linked recessive disorder problems, okay, and also in the transcription, translation, lack of a lot of concepts we are going to cover. We have 2 hours in our hand. In these 2 hours, we are going to cover these 3 chapters, and moreover, each chapter, how many concepts we should thoroughly prepare, also today we are going to understand. Am I clear? Are you ready, all of you? Look at here. First chapter, principles of inheritance. In principles of inheritance chapter, which all the topics we should cover first? Everyone, student, keep in mind, principles of inheritance chapter is a big chapter in genetics and evolution unit. So in this principles of inheritance chapter, which all the important concepts are there that you should 100% finish? Look at here. First one, Mendel laws. Oh, here Mendel laws we should finish first. Now tell me here, law of dominance, law of segregation, law of independent assertment. First all 3 laws, and introduction part of Mendel also, okay, introduction part of Mendel, and on the pea plant, which plant is that? Pisum sativa, and contrasting traits also, which one is dominant, which one is recessive, and all the laws of Mendel. Next, come to here, monohybrid cross, dihybrid cross, especially this test cross. We will discuss for what purpose we will do the test cross, okay, to identify the unknown genotype of we will do the test cross. We will discuss the test cross also, and based on the test cross, various questions also we will discuss. And after that, incomplete dominance, co-dominance topic, especially examples of this one. Sometimes they will give you a question in the form of a red flower of is snapdragon or antirrhinum, Andalusian fowl. This type of question will come. So it is not follow the Mendelian laws. So this is what? Non-Mendelian cross. So incomplete dominance, co-dominance, multiple allele. Multiple allele is where? Blood groups. That also you should cover first. After that, chromosomal theory of inheritance. Sutton and Bovary chromosomal theory of inheritance. Very, very important. This topic also we should cover. Next, linkage and recombination. Linkage and recombination, T.H. Morgan on Drosophila melanogaster. This recombination and linkage, they will give you the particular location of the genes, A gene, B gene, the distance, and what is the recombination ratio and what is the linkage ratio they will give. So this topic also we should cover in this class, and especially when you are preparing for your this final exam, this topic also you should 100% touch. Next one, pleiotropy, polygenic inheritance. These are the very simple topic. Pleiotropy, polygenic inheritance are very simple topic, and this topic also you should cover with examples. Now here, sex determination, one of the important concept in principles of inheritance. Sex determination, male heterogamity, female heterogamity, ZZ ZW, ZZ, Z0, XX, X0, XX, XY, all 4 type of sex determination, including examples, you should finish. Next, after this one, mutation and disorders plus pedigree also here. Pedigree also, mutation, especially what type of mutation? Gene mutation and also chromosomal mutation. We will discuss these all the topics from where? Principles of inheritance. Shall we go to the first question? The first question I am going to discuss. Look at the first question. First question is, what is the first question here? First question, look at here the first question. Given pedigree of a family, one pedigree is given. This is a 3-mark question. Look at here, they given A, B, C, 3 questions are given here. right? This is a 3-mark question. One pedigree is given. In this pedigree, look at here, in this pedigree, father unaffected and mother also unaffected. The F generation is, this is a F generation is unaffected, and irrespective of sexes in the F1 generation, here it is daughter affected here and son also affected here. Can you guess what type of heredity, what type of disorder is this? What type of disorder is this? Any one of you? Right? Autosomal recessive. Hafiz, absolutely autosomal recessive. Everyone keep in mind, if you are considered this is a sex linked recessive, chalo, we will assume it is a sex linked recessive. Tell me here, this is a sex, if it is sex linked recessive, now here father is normal. If just assume the mother is carrier. If the mother is carrier in the F1 generation, this daughter should not show the disease, right? Daughter should not show the disease. Am I clear? Are you with me? So this is not sex linked recessive. So what type of recessive is this? Autosomal recessive. So first question is what kind of inheritance can be observed in the given pedigree? The given pedigree inheritance is autosomal recessive. What are the examples of autosomal recessive? We learn sickle cell anemia, phenylketonuria. Am I clear? Cystic fibrosis, thalassemia. And these all are autosomal recessive disorders. Am I clear? Now look at here, F generation is not affected, and moreover I told you this one in our theory class also we discussed, when parents unaffected and children are affecting, this is a characteristic feature of recessive disorder. Recessive disorder. Now look at here, next, write the disease which exhibit this type of pedigree. Any disease you can write. Disease they are asking. What is the disease? What is the disorder they are asking? Autosomal recessive in phenylketonuria. Or you can also write the mutant gene of what? Sixth position of hemoglobin. What is the sixth amino acid? It must be glutamic acid is replaced by the valine. That also you can write. Sickle cell anemia. So I am writing here, write the disease. Sickle cell anemia is also one of the autosomal disorder. Remember student, when pedigree questions come, carefully look at which generation affected, which generation did not affected. Am I clear? Daughter and son both are affected, parents affected or not, all the 3 generations affected or not. Carefully look at the pedigree. Am I clear? This can become for the 3-mark or directly 5-mark also, or sometimes they give a case study also on the pedigree chart problem. Look at the last one. Last one, any one of you can guess, any one of you can answer me. The last one is, what is the last one? A normal vision woman whose father colorblind. Can you please tell me the karyotype of this the genotype of this woman? Genotype of this woman, any one of you? Sania, Alicia, can you tell me the genotype of this woman? A normal vision woman whose father colorblind. Whose father colorblind means, shall I call this woman is carrier? Shall I call this woman is carrier? Right? Right, good. Now tell me here, I can write here. What tell me here? A normal vision woman, XCX, she must be a carrier. She must be carrier, right? Now marry a normal vision person, X and Y. Okay, now let me take the gametes here, XC and X. Here it is X and Y. Okay, look at the first daughter. What is the first daughter? XCX, she is a carrier. So she is unaffected girl, unaffected daughter. Fine. Now what about this one? What about this one? XCY. So XC and Y, affected son. We know, we have already discussed, sex linked recessive disorders are transferred from mother to sons, not from father to son, because father is contributing Y chromosome. I hope you are following me. I hope you all are following me. Right. Now tell me here. Tell me here. Next one is X and X. Now here it is a normal girl. Okay. Last one is called X and Y. So shall I call 50% of sons are affecting? Shall I call 50% of are it affecting? Am I clear? Till here everything clear? Now answer me. Look here, what the question asking. A normal vision woman whose father colorblind marries a normal vision man. What would be the probability of her son's daughter to a colorblind? First half. Sir, what is the answer of this one? Sir, 50% of sons are colorblind and none of the daughter. 0 daughter affected. How many of you follow me? Yes, very good. 0 daughter. Hafiz, absolutely correct. Can you look at the other half of the question? Can you look at the other half of the question? Explain with the help of pedigree chart. Explain with the help of pedigree chart. They are asking. Remember, if this type of question come, it's very simple. So we can write like this. Now shall I write like this? How? This is mother and this is father, unaffected, right? Unaffected, because in the question they given a normal vision woman whose father colorblind, but the husband is normal only. So they have, now I'm writing here one girl, one boy, one girl and one more boy I'm writing here. So shall I write 50% of the sons are affected, so 50% sons are affected, rest of all are normal only. So pedigree, in the form of pedigree we are showing. Are you with me? Are you following me? Everything clear, Sania? Clear? Alisha, everything clear? So this type of question, 3 mark pedigree based question will come. You have to find out it is autosomal dominant or recessive or based on this one, characteristic feature, disease name, anything can be possible. Anything can be possible, student. Chalo, next question. Question number 2. Question number 2, I am going to look at here everyone. What the question they are asking here? Define and design test cross. Define and design the test cross they are asking. Test cross is that cross to identify the dominant of the genotype or recessive, heterozygous or homozygous. That means unknown genotype. We are identifying unknown genotype. For this process, can any one of you tell me with which parent we will cross? With which parent we will cross? The recessive parent, right? With the recessive parent we will cross, and based on the ratio we will get this particular genotype is homozygous or heterozygous. Let me take one small example. They are asking design also they are asking. So here one unknown genotype is capital T, homozygous, one is the recessive. Oh, sir, one is the heterozygous, one is the homozygous, one is heterozygous. Chalo, answer me. We don't know this genotype. We don't know what is test cross actually. To identify the unknown genotype, we will do what? Test cross. So tell me here, this is the unknown genotype. I am doing a test cross. I am doing a cross with the recessive parent here. Now based on the ratio, so in the F1 generation, I will get all tall plants. When I get all tall plants, I can write here 4:0. Based on this ratio, I can tell this is a homozygous. Because our known genotype characteristics did not repeated, did not form in the next generation. What is our known one? Our known one is recessive, but there is none of the plants are recessive, none of the plants are dwarf. Are you with me? So this is test cross. Come to this side. Come to this side. So what is the other parent now here? Now cross with small t. This time you will get 50% of tall plants. Am I clear? 50% of short plants or dwarf plants you will get. So 50% of your known plants are coming. That means your unknown must be heterozygous. Are you with me? So test cross is that cross if you are crossing that unknown genotype with what? Tell me, recessive parent. Right? So this is define, and you have to design a particular test cross. You can take any of these two examples, or you can take both examples also, you can write. Now next question, question B. That means it is not ABC in one question, beta. I'm selecting the questions in the form of ABCD. This is completely different question. Look at the question. You are given a red flower bearing pea plant. Look at it carefully. Look at here, red plant bearing pea plant. Answer me. In the pea plant, red flowers are the dominant. Red flowers are dominant, right? Now, red flower bearing snapdragon. Can you please tell me here? Snapdragon means it is an incomplete dominance, right? Snapdragon means it is an incomplete dominance. Yes or no, students? Right? So carefully look at here in normal cross. If you are writing directly the snapdragon red flower also dominate, it will be wrong. It will be wrong. So carefully look at here. The pea plant red color flower is a dominant. Done. Now snapdragon plant. How would you find the genotype of these two plants? With the help of cross, respect to the color of flower is given. That means you need to identify the genotype. Shall I call this a type of test cross? Shall I call this a type of test cross? Alicia, Sania, and who is this? Shadi is a test cross. So how can I solve this question? Look at here. I can solve this question. Look at here. Here it is simple. capital R, I am cross with small r, recessive one. So I will get here capital R, here it is small r. In the F1 generation, capital R, small r. In F1 generation, red color flower comes in pea plant. But if you are doing the cross with the snapdragon, can you please tell me what type of color will come? If we are doing this test cross in the snapdragon red color flower plants, what type of genotype, what type of phenotype comes? Any one of you? Very good, absolutely. What is the genotype? Will come? Capital R, small r. But here it is, the capital R does not completely what they call dominate over small r. Because of this reason, it is not showing the law of dominance. Because of it's not showing that what they call dominance, we can say incomplete dominance. So I will write, look at here, capital R, red flower, and small r, white flower. When I cross capital R, small r, F1 generation, this is F1, pink color forming, intermediate color is forming. So when intermediate color is forming, this is incomplete dominance. That's why I'm telling you, student, when you're going to the examination hall, if a snapdragon, Andalusian fowl, antirrhinum, this type of examples come, be careful. Do not follow the Mendelian clause. They will not follow the law of dominance. Am I clear? So this is this question. Next, come to C. Compare the mechanism of sex determination in human. This is very, very, very important. Carefully look at and understand the question. Sex determination in human being with the bee, honey bee. Now let me write here. We have already discussed this one. Here, human being sex determination is XX, XY type of sex determination, and human beings are diplontic organisms, right? Let me take here one paper. Look at here. Human sex determination is XX, XY type of sex determination. So I'm writing here 22 plus X chromosome of one egg, and here 22 plus X chromosome of sperm, or 22 plus Y chromosome of sperm, because human is male heterogametic. Fine. So here this egg will fuse with this one, so 44 plus XX, this is female, or this one is male. Yes or no? 44 plus XY. Simple human sex determination. But hold on. In bees, if you able to recall bee hive, honey bee, diplontic chromosome number is, can any one of you tell me? 2, and diploid number of chromosome of honeybee, any one of you tell me? What is the number of chromosome in the diploidic number of chromosome in honeybee? Anyone? Alisha, Sania, Khalid, anyone? Sana, Romila? 32. 32 is the diploidic number of chromosome. So here what happened? Honeybee will produce 2 type of egg. So these eggs are meiosis division, the egg which is contained n number of chromosomes are 16. This haplontic egg, if it is fused with the haploidal sperm, both gametes are fused to form a zygote. This zygote is diploid. This diplontic fertilized egg only become female bee. Remember, this zygote will develop into female bee. Remember, all of you. What about the next type of eggs? Any one of you guess? Sir, there is a next type of egg which is a 16 chromosome haploid. Without fertilization, they become an embryo. They become a zygote. And this zygote is become new individual that is called male bees. So male bees are haploid. This process called, any one of you tell me the process of a development of embryo without fertilization? Parthenogenesis. Yes or no? Very good, absolutely good. Parthenogenesis. So shall I call the honeybee sex determination is haplodiplontic? Honeybee sex determination is depending on the sperm fertilizing or not. If sperm fertilize, it will be female. If sperm is not fertilized, the egg is not fertilizing, it will become a male. Am I clear? No doubt. Remember, sir, you could have asked one question. Sir, this 16 chromosomes, the males are haploid. That means males are having the 16 chromosome. So how males are going to produce the sperms? Any one of you? Males are going to produce the sperm by dash division. Males are going to produce the sperms by dash division. Meiosis or mitotic? Meiosis or mitotic? Remember, mitotic division, not meiosis. So I am writing here, it is what? Mitotic division. Everyone keep in mind. So sex determination in human being depending on XY chromosome. Sex determination in honeybee depending on fertilized egg or not. Come to the next one. Look at here, all of you. Sex determination in human with the respect to honeybee, with the respect of chromosome number. Okay, fine. Now come to the second. How is the gamete formation comparable with the above two cases? Gamete formation. Sir, gamete formation in case of human being is only by meiosis division. Are you with me? Are you with me? Only meiosis division in the spermatogonial mother cell. We discuss in testis. We discuss in the ovary. Yes or no? Oogonia, spermatogonia we have done. That diploidal cells undergo meiosis 1, meiosis 2 to produce the haploidal gametes only. But what about the honeybee? What about the honeybee? Sir, in honeybee, the female gamete is producing by meiosis. Go back here. Look at here. Look at the female gamete is by meiosis division, but male gametes by the mitotic division, sir. Remember, all of you, sex determination in case of honeybee and the gamete producing of honeybee. Am I clear? Are you with me? Now, next question. Look at here. This is C question. Look at the D question. Look at the D. I will be silent. Can any one of you tell me the D question? Name the type of cross that would help to find the genotype of P in a violet flower. I will be silent. I am looking your comments. Come on, answer me. Hi. Flip flop. Welcome. Chalo, can you answer this one? Name the type of cross would be help to find out the genotype. Sir, it is a simple test cross. Absolutely, test cross. Remember, all of you, monohybrid, dihybrid, test cross, and various questions from test cross, incomplete dominance also. Till here everything done? Next question. Next question. Look at the next one. This type of questions will sure. This type of questions will come carefully. Look at the questions. Point one of the important concept in principles of inheritance is mutation. I have a question. Before I go to this question, can any one of you answer me who given the term mutation first? Who proposed the mutation theory? Who proposed the mutation theory? Any one of you? Very good flip-flop. So, but first you answer my question. After that, we will go this one. Who proposed mutation? Mutation, very good, absolutely. Hugo de Vries. Hugo de Vries also said that saltation. We will discuss that also. Saltation or mutation. Look at the first one. What is point mutation? Give an example. One example. Point mutation is that mutation a single base pair altered. Maybe insertion or deletion or it altered. We can say it is altered. Let me take one small example. Here I am writing GAG. What is the complementary here? CTC. And let me write here, this is a 5 prime, 3 prime. This is what? 3 prime and also 5 prime. Let me write here, this is 3 prime, 5 prime. Okay. Now, this is a template strand. Template strand undergoing mRNA transcription to form, tell me, GAG, 5 prime, 3 prime, mRNA codon. This is codon. It give what? Tell me, glutamic acid. Glutamic acid, fine. Now answer me, student, what is point mutation here? Sir, here one nitrogen base is going to mutate to form what? To form what? Tell me here. I can write G. Can you please tell me? A is replaced with T, right? GTG. GTG. So I can write what here? It is GTG. I am writing here, it is what? CAC. Now what is the mRNA here? GUG. Instead of GAG, GUG is forming, GUG is valine. So point mutations are that mutation, one single base pair altered. One place. And remember all of you, before point mutation, after point mutation, the codon is good. No problem. Let me take here, before this one, after this one, the codon, the DNA is good. One place only a change takes, one base pair only altered. Such type of mutation called point mutations. This is the example of sickle cell anemia. This is the example of sickle cell. Are you with me? Yes, absolutely. Phenylketonuria also, sickle cell anemia also, right? Am I clear? Now give one example. You can write any example of this 4 out of the 6 cystic fibrosis. You can write sickle cell anemia. You can write. Next, point mutation. Remember, point mutations are frame shift mutation also will come, student. Frame shift mutation, missense mutation, nonsense mutation, silence mutation. There are different type of mutations are there. Next, come to the second one. Two heterozygous parent are crossed. If two loci are linked, what would be the distribution of phenotypic features in F1 generation for the dihybrid cross? Carefully read the question. Carefully read the question. Hafiz, Alisha, Shadi, careful, Dharani, carefully read. They are asking dihybrid cross. Look at here. Two heterozygous parents are crossed. Let me write here. Two heterozygous means, shall I take like this? Capital T small t, capital R small r, cross with capital T small t, capital R small r. Two heterozygous. Two heterozygous. If this is normally following the dihybrid cross, in next generation, what is the phenotype ratio? What is the phenotype ratio? If normally it is crossing, we will get 9:3:3:1. Am I clear? Correct? Normally we will get 9:3:3:1. But read the question carefully. The genes are closely present, linked genes they are asking. To understand this question in better manner, look at here, all of you. Carefully look at here. Heterozygous, capital T small t, capital R small r, cross with capital T small t, capital R small r. Everyone carefully listen here. I am taking this homologous chromosome. This is homologous chromosome. Here I am writing capital T. The next allele I am writing here, it is a small t. Here it is capital R. Here I am writing small r. Loci. Okay. If they are present like this, they are apart. That means they are distant. They are far apart. They will segregate. They will form a new gametes. 4 different type of gametes can come. But they are linked genes they are given. So I can show if it is a linked gene. This is homologous chromosome. Capital T here. Now look at here. This is small t here. Here I am writing capital R. Here I am writing small r. The distance between the genes is reduced. Yes or no? So in that time, everyone keep in mind, they will not independently assert. Are you with me? Independently they will not assert. So what happened when this homologous chromosomes aggregate? When homologous, we know anaphase 1 of meiosis 1, 2 homologous chromosomes will be moved towards the pole. They separate. So look at it. This chromosome will separate to form one gamete. What is a gamete? Capital T and small t small r, one gamete, and this will separate to form one gamete, capital T and capital R. That means 2 type of gametes only coming. And that also capital T, capital R, small t, small r. So parent characteristics only repeat, right? Look at here. Capital T, capital R, small t, small r, capital T, capital R, small t, small r. So here also I am writing capital T, capital R, small t, small r, capital T, capital R, small t, small r. So if you cross this one, you will get capital T, capital T, capital R, or small t, small t, small r, small r. There is no new variation. Recombinants will not form. You got my point? Recombinants will not form, because if you read this question, they are giving to us, they are heterozygous parents are crossed, loci are linked. So remember all of you, when 2 genes are linked, they will not segregate, they fail to segregate, recombinants will not form. So parent characteristics only repeat. Majority of parent characteristics only repeat. This was given by T.H. Morgan in his Drosophila experiment. How many of you got this? Till here everything clear? Fiplov clear? Asil clear? This one? Sania? What I said clear? Because they are closely present linked gene, so they will not segregate. If they are not segregating, they cannot make a new recombinant, they cannot make a new gametes. So when the new gametes are not formed, no recombination, no recombinants. Till here everything done? Clear? Right, next one. Look at the next one. Mention any two autosomal genetic disorder with their symptoms. They are asking with symptom. I will tell you one symptom, you just tell me. Accumulation of phenylalanine pyruvic acid in brain to cause mental retardation. Can you tell me the name of the disorder? Can you tell me the name of the disorder? Sir, what's the phenotype distribution? Right, phenotype distribution is what? Look at here. Tall, round, tall, round. Look at, I'm taking a parent here. Tall, round, heterozygous. In the next generation, tall, round or wrinkle short. Parents only will come, right? Parents only will come, right? Next. Next one is what? Now, this is autosomal recessive. What I said? Accumulation of phenylalanine pyruvic acid in the brain to cause based on this information we can tell what it is. Phenylketonuria, autosomal disorder. Sickle shape of RBC is biconcave shape of RBC converted to sickle shape. So what is the answer? Sickle cell anemia, sir. Sickle cell anemia. Sickle cell anemia also autosomal I said, and phenylketonuria. This important amino acid phenylalanine does not convert into dash. Phenylalanine does not convert into tyrosine. Am I clear? Tyrosine. So we can write two genetic disorders with what? Symptoms. Fine. Next. Now carefully look at here what is asking. A cross between two tall plants result in the offsprings having the dwarf plant. Two tall plants resulting in the, if you are crossing two tall plants, result the formation of dwarf plants. I will be silent. Any one of you can do this one? Any one of you tell me what type of parent genotype here? What is the genotype of the parent? Here, what is the genotype of the parent when both are heterozygous? I already given answer. Oh my god, I already given answer. Both are heterozygous, right? If both are heterozygous, only there is a chance of the short plants can come, right? Am I clear? Chalo, let me take here. If I take capital T cross with the capital T, in this one, is there a chance of a short plants in the next generation? In this one, is there a short plant? Is there? So no. Sir, in the question, they given two tall plants, but they did not mention heterozygous, homozygous. They are asking you to guess that. Look at here, the genotype of both parents they are asking. Genotype of both parents are heterozygous, sir. Because of heterozygous, F1 generation is what? F1 generation, next generation they are showing half of the 25% of short plants are coming. Remember, when this type of question come, carefully read the question. If the two tall plants are homozygous, no chance of what they call short plants are come in the next generation. Are you with me? These are a simple question from dihybrid, monohybrid, test cross questions. Now, come to here. What is the question? The pedigree chart given below shows a particular trait which is absent in the parent but present in the next generation, irrespective of sexes. Draw your conclusion on this pedigree. What type of pedigree is this? What type of pedigree? Parents are unaffected, irrespective of sex, girl and boy, both affected. What is this? Right, what is this? Answer me. So this is called what? Autosomal recessive condition, because the girl, irrespective, girl is affecting, boy also affecting. This boy is affect. That means I can write here. Shall I write the genotype here? Genotype of all one. So look at here. Shall I write capital A, small a of parents? So this must be what? Capital A or capital A, small a. That's why this girl is not showing. So what about this one? And what about this one? Shall I write it is a small a, here also small a? Sir, we know autosomal recessive condition, if the two recessive genes are present only, that means the homozygous condition only it will repeat. That means it will express. Yes, so here it is, it could maybe capital A or capital A small a. Here it is capital A or capital A. So sometimes they give pedigree, they ask the genotype, student. Sometimes they give a pedigree, they ask the genotype, you can identify the genotype. But here, based on this small irrespective sexes in the next generation, I can decide, I can tell this is autosomal recessive disorder. Next, look at this question, my dear student. Look at this question. In pea plants, tallness is dominant over the dwarfness. Tallness dominant over the dwarfness. Okay, all of you carefully. Kirtraj, careful, look at here. What I'm telling. Isra. And red color flowers dominant over the white flowers. Red color dominant over the white. When a tall plant bearing red color flowers was pollinated with the dwarf plant bearing white flowers. Student, in this question, two parents are given, one parent genotype we know. Tell me which parent genotype we know? Which parent genotype we know? There is only one chance is there. Any one of you, I will check. I will check. Any one of you tell me. There are two plants. Tall plant bearing red flowers and dwarf plant bearing white flowers. One of these two parent genotype we know better. What is that? Any one of you? Any one of you? These two, one parent genotype we know. Second, very good, flip-flop, absolutely correct. So what is the answer? What is the genotype, the second parent? Any one of you tell me? So I can tell that the genotype is small t and small r. Shall I write? Here it is, white flower they are asking, okay? White flower, so shall I write here it is small t, small w. We know this one. Done. Now take here one paper. Now I am writing here small t, small w. It is what I can write, short and white flowers, right? Now it is crossing with tall and red flowers. Fine. Come to the question. Student, carefully look at here. What I'm doing. Now, the different phenotype groups were obtained. Wait. Different group of plants obtained. Come to here. Come to this one. Here I am writing. If this first parent is capital T, capital R. Okay. So here I am taking. Okay. Now here I am taking short small t. Let me take small r. Answer me, in the next generation F1, different type of plants, there is a chance is there? Tell me, in the next generation, is there a different type of phenotypic plants and genotypic plant chances there? Sir, no. One type of plants can form. One type of phenotype plants can form because this is a dominant one. So what type of plants can form? Capital T, small t, capital R, small r in F1 generation 16 by 16 form. But the question tells us the different type of were obtained. Can you guess? If it is a different type, our first parent is homozygous or heterozygous? Our first parent, that means this red color flowers and tall plant is homozygous or heterozygous? Any one of you? Absolutely. That's why I'm telling you, student, read the question carefully. If it is a homozygous tall and red flower, there is no chance of obtaining different phenotypes. Different phenotype, there is no chance. So I'm going here. What I'm doing here, look at here, all of you. So this is the one. So we already decided, we could discuss this one. This is not a correct one. Fine. So it must be, first parent must be capital T, small t, capital R, small r. Right? So this is a test cross. Look at your test cross. Recessive parent we are crossing. Fine. What are the genotype of offsprings? What type of plants can come? Look at here. So here, can any one of you tell me how many different type of gametes can form? Capital T, capital R, capital T, small t, capital R, small t, small r. What about from this parent? Small t, small r, small t, small r, small t, small r, small t, small r. First one, what is the first one? Tell me. Capital T, small t, capital R, small r. Second plant, third plant, fourth plant. So here I can write, tell me, capital T, small t, capital R, small r. What is this? Tall red flower. Fine. Come to the second one here. Capital T, small r, small r. Can you tell me what is this? What is the phenotype of this one? Tall white, shall I write? Tall white, shall I write here? Right? Now I'm writing it capital T, small r, small r. I'm writing here it is tall white. Can you please tell me the third one? Any one of you tell me the third one? Any one of you tell me third one. Flip flop, what you're doing? Answer me. What is this one? Small t, capital R, small r. So tell me, what is this one I can write here? Here I can write short red. Look at here, short red. So small t, capital R, small r, short red one, because dominant gene is there. Red is a dominant gene. Last one, any one of you? Last one is a recessive. So we can write small t, small r, for plant. So I can write small t, small r is short white, sir. Short white. So shall I call this one is a 4:4:4 ratio or 1:1:1 ratio? 1:1:1 ratio. So look at here. Come to the question. What the question is telling. Now, number of obtained against red tall 138, tall white 132, dwarf red 136, dwarf white 128. Mention the genotype of two parents. First two parent genotype is capital T, capital R, small r. Second parent is small t, small r. Next term, or four offsprings type they're asking. Four offsprings type we wrote here 1, 2, 3, 4. How simple. If you understand the question, if this parent is not homozygous, you will get full out of full marks. 100% you will get full out of full marks. If you are directly thinking, the two parents are homozygous, capital T, capital R plus small t, small r, no, you will not get. Till here everything. Is this question clear? Shadi, clear? Allah, flip flop, clear beta? Kirtiraj, everyone clear? This question clear? This is a test clause. 1:1:1 ratio is coming. Any one of you? Come on, answer me. Till here everything done? Yes, very good. Right, but the number of given is not, not same, but it is a type of test clause. Look at it, almost is equal, almost they are similar. Whatever it is given, it is number is similar. So forget about this number. Look at this phenotype, flip flop. Look at this genotype. So four different type of genotype is coming. That means the heterozygous parent. It's a 1:1:1. Am I clear? Now, next one. Till here everything clear? Done? Flip flop, you got my point? Done. Very good. Now, this question. Mark my words, all of you. Linkage, recombination questions. My God, I'm really telling you. Carefully prepare and go. Look at the location of gene, recombination type, recombination ratio, percentage, parental percentage. Carefully. Look at the question what I'm teaching now. All of you, pay attention here. The given figure below, answer the question is given. Cross A, cross B. Fine. Yellow, white, wild type. Okay. Now this is a linkage and recombination given by Mendel. Look at the first question. In dihybrid cross, when would be the proportion of parental gene combination B much higher than the non-parental type? I will be silent. Answer me. I will be silent. You have to answer me. Parental gene or parental combination much higher than the non-parental. What does it mean? Come on. What does it mean? Answer me. Sir, that means that two genes are linkage genes. Am I clear? Yes, more link. Absolutely correct. Who is this? Ayan, absolutely correct. Sir, linkage is more. Distance between the gene is less. Genes are closely present. So because of that reason, parent combination is coming. Am I clear? Wait, look at here. as experimentally shown by the Morgan and his group. Yes, experimentally T.H. Morgan proved that instead of getting 9:3:3:3, 9:3:3:1, I'm sorry, he got 8:8. That means total pay half 50% parent character, 50% parent character. That means 98% of approximately 98% some change parent characteristic only. No recombination. Why no recombination? The body color and eye color of Drosophila genes are linked gene and exclusively they located on X chromosome. Where they present? X chromosome. Got my point? Look at the second question. If two genes are located far apart from each other chromosome, how frequency recombination will get affected? Carefully look at here. If two genes are far, how frequency of recombination will get affected? Sir, the linkage is inversely proportional to what? Recombination. When genes are far, when genes are what they call far apart, so distance is more, they will segregate. Law of segregation takes place, they will separate. So then separate, different type of gamete they form. So different recombination can come. Recombinants will form. Do you remember 9:3:3:1? These 3:3 are recombinants only. Recombinant. So that means it will affect. What will affect? Frequency of recombination will affect. When will affect? When genes are far. When genes are far, that's why, do you remember, if you remember, Hardy-Weinberg equilibrium, one of the factor which affecting the Hardy-Weinberg equilibrium is recombination. How many of you know this? How many of you know this? One of the factor which affecting the Hardy-Weinberg equilibrium is recombination. Yes or no? Allah, Sania, Kirtraj, Hafiz, am I clear? One of the factors is recombination. So recombination from the new characteristic is coming. Am I clear? Keep in mind. So it will affect. What are true breeding lines? They're asking. Any one of you tell me what are true breeding lines? True breeding lines means, any one of you? A plant which is self-pollinating for many generations. So it will become a homozygous for that trait, that character. But if you able to recall sexual reproduction in flowering plant chapter, continuous autogamy leads to the process called inbreeding depression. Do you remember this? Inbreeding depression. But here what happened? The true lining plants, or we can say true breeding line plants, are that plant, the plants which are self-pollinating for generations to come homozygous condition. When this homozygous condition, superior characteristic will be go to the next generation if you pollinate it with the other superior plant. That's called what? Tell me. True breeding lines. Now, study the inheritance pattern, the traits of plant. True breeding plants are self-pollinating for many generations and their homozygous condition to carry this trait to the next generation. Understand? Or this question number C, or question, any one of you answer me. Any one of you answer me. I will cross check. Any one of you, come on. Ayan, Kitraj, Sania, anyone, Allah, come on, look at here. If a frequency of parent form is higher than 25% of dihybrid testosterone, parental form is higher than 25%, what does that indicate about the genes involved? Be a part of a question again. Which one, Sanjay? So genes are closely linked mostly. Yes, correct. So which one is this, Sanjay? Which question? Right, look at here. When more than 25% means what are the genes are linked. That's why more than 25% of parent characteristics only repeating. Am I clear? I hope you all are following me. Very good. Absolutely correct. Linkage, recombination, only one thing remember, when genes are there, you can get a question. This is gene A, gene B, gene C. The distance between gene C, gene A is 10 units, 5 units, 3 units. They will ask you different type of what? Questions on this one. Done? Question number 5. Okay, we'll go this one. Question number 5. Now, question number 5. Which one? Question number 5. Question number 5, A, B, C. Which one? Question number 5, A, B, C. Which one? Closely linked, sir. We will do this. Okay, next question. One of very important. Sanjay, don't worry, I will explain you. Okay, B you are asking. Okay, I'll explain you this one. This question, beta chain of polypeptide, normal hemoglobin. Carefully read what exactly the question is given. This is a very important and complicated one. You have to know about the genetic code. A codon for 6th amino acid is GAG. Codon is, let me write here, what is given? GAG. Let me write here, this is, what is that? Where did it go? Now look at here. Codon, what is given? 6th amino acid is GAG. Okay, 6th one is GAG. Fine. This is A mutation. This is codon. Can you please answer me? GAG code for what? Which amino acid? Glutamic, right? Glutamic acid, right? So GAG code for glutamic acid. Fine. Okay, careful, look at better. This 5-mark question. Now look at here. Now this one, GAG mutant to GAA. Come back here. GAG, this one is mutant to form GAA codon. Now carefully answer my question. If you look at the genetic code, GAA also code for glutamic acid. GAA also code for the glutamic acid. If you are not believing me, cross check. GAA also code for the glutamic acid. Am I correct? Am I correct or not? Answer me, any one of you. Lots of biology. Come on, answer me. Yes, tell me. Yes. GAA also code for the glutamic acid. That means if a mutation takes place also, no problem, because the same amino acid coming on the 6th position of hemoglobin. Are you with me? Are you with me? You got my point? Now come to previous one. So here, what is given here? GAA. As a result, the mutation A. Mutation A is GAG to GAA. Done. GUG result in the mutation B, hemoglobin. What is here? Tell me. Mutation B is, look at here, this one, mutation B. Careful, look at here. GAG mutant to form GUG. Can you please answer me GUG code for? GUG code for? GUG, just now we discussed valine. Valine, absolutely right. GUG code for valine. Fine. So that mean, go back here. That mean problem with the mutation B. Am I clear? Problem with the mutation B. Mutation A is okay. Mutation A, GAG mutant to GAA, no problem, because both are code for the glutamic acid only. Done. Now hemoglobin structure did not change result of mutation A. Hemoglobin structure did not change, whereas hemoglobin structure changed because of mutation B leading sickle cell of RBC. They given this figure. If you don't know GAA also code for the glutamic acid, you will not answer this question. No, you will not answer this question, I'm sure. If you don't know GAA also code for the glutamic acid, you will not. Look at here. Explain given reason how could mutation B change the hemoglobin structure and not mutation A. Sir, mutation A, it is GAA also code for the glutamic acid. So no problem, the sickle shape of RBC, the biconcave shape of RBC remain in the form of biconcave. Done. But in case of B, it is converting into valine. So valine is replacing there. Done. Next. Or write the genotype of both parents who have produced the sickle cell anemia. Sir, sickle cell anemia is coming when parents are heterozygous. So I am writing HBA, HBS and cross with HBA, HBS. Done. So there are 25% of sickle cell anemia, what they call offsprings. Next, write the genotype of an individual who is carrier sickle cell anemia gene but apparently unaffected. That means I can write HBA, HBS, carrier person unaffected. Now an individual affected with disease, is HBS. Am I clear? Till here everything done? No doubt? Any one of you clear? So we need to no need to learn all the codons, but there are few codons are there. You must remember, like a stop codon, start codon, okay, and also AUG and UGG. Few are there. You should 100% remember that. So this is the answer of this question. Keep in mind, if you don't know GAA also glutamic acid, you cannot answer the first question. No. Next, come to the next question. Next question, very easy question. First one, can anyone of you answer this one? What is the syndrome of this one? If 21st trisomy is there, 21st trisomy I can call what? Down syndrome. My dear children, before 16th you must prepare Klinefelter, Down syndrome, Turner syndrome thoroughly. Aneuploidy, monosomy, trisomy. Here I am writing what? Karyotypy of child shows the trisomy number 21. Identify the disorder and state the symptoms such as linkage to be exhibited in this case. Here, so likely to be what they call exhibited in this case. This syndrome is called a Down syndrome. The characteristic feature you can write: fur, tongue, partially open mouth, okay, palm, seed. This all the characteristic feature of Down syndrome. You can write this Down syndrome, what they call trisomy. Question B. If father and son are both defective red-green color vision, father affected, son also affected. Clearly read the question. Father affected, son also affected. Now likely that the son inherited the trait from his father? Question mark. Is this inherited from the father? I need answer. Any one of you tell me, if son affected and father also affected, this son receiving this genetic problematic gene from the father? I need answer. Any one of you? No, absolutely. Why, sir? Color blindness or color blind is not transfer from father to son. It transfer from mother to son. It transfer from where? Mother. So though the father affected and son affected, it is not, the son is not receiving any defective allele from where? Father. He received defective allele from, it must be from where? Mother. Remember, X-linked recessive is not transfer from father to son, it transfer from father to daughter. Am I clear? Father to daughter. Next question. A cross is made between the different heterozygous pea plant contrasting flower position. How many positions are there? One is called exile, one is called what? One is exile and one is called what? Any one of you? Terminal. Which one is dominant? Exile is dominant, terminal is recessive, right? Find out the position of the flower in F1 generation. So simple, I am taking like this. Capital A, I am writing this is exile position. Sanjay, can you please answer me? Small a, I am writing terminal position, right? Terminal position. So in the F1 generation, I can write small a, capital A, small a. Exile is the one which is F1 generation. Clear? Everything clear? Kitraj done everything here. Exile, fine. So F2 generation I can write capital A small a cross with capital A small a. I can get capital A exile and terminal. Am I clear? Is this question is clear? Very simple question. You can easily write the answer. Look at the last one. F2 generation and computed related what they call fractions of various genotype in the F2 generation. Various genotype, 3 different type of genotypes can come: homozygous, heterozygous, recessive one. Done? Homozygous, look at here, homozygous, heterozygous and also recessive one. I am going to the next question. Look at here. I think this is the last question of principles of inheritance. The important concept coming, molecular base of inheritance. Look at here, everyone. Identify A, B, C, D, E and F in the table given below. This is a 5-mark question. Look at the first one. Down syndrome, trisomy 21, 1 and 2 characteristic feature. 2 characteristic feature. Any one of you? 2 characteristic feature of Down syndrome. Sir, partially open mouth and partial what they call we can say partially open mouth, furrowed tongue, this all a round headed face and palm seized. These all are the characteristic feature of low IQ. These all are what they call trisomy. What is the trisomy? Down syndrome. Male or female? This one, can any one of you answer me? Male, female, male or female, or both? Both or male or female? Allah, this Turner syndrome persons are only male or only female or male, female, both? Yes, absolutely, absolutely correct. There is a chance of both can be affected here. So you can write what? Tell me here. Male and female, both can be affected. Fine, done. What about the second one? C is given here. Syndrome XXY. Sir, this is Klinefelter. Klinefelter syndrome over a masculine development, gynecomastia, and this D is what? Tell me. D is only, I think, only females, I think, right? D is only female. Klinefelter, yes sir, yes Azim, correct. Males, not female, male. So I'm writing here this what? XX chromosome, it is a male. This is a trisomy. Turner syndrome, 3. Turner syndrome and X45 plus with XO. Right? Next we can write what? Tell me here. 2, 3. So shall I write that E, 1 and 2, poorly developed or rudimentary ovaries, undeveloped ovaries, no secondary sexual character, fertile female, no menstruation. Am I clear? These all are the characteristic feature of Turner syndrome due to the lack of one X chromosome. What about F? What about F? Yes, female, absolutely. So we can write, Sania, what is this, Azim? Here it is female. That means I need answer. I need answer. Now answer me. Here it is. These 3 are there. Can you please tell me out of these 3 which is the trisomy? Which is the trisomy? Sir, there are 2 trisomies are there. One is autosomal trisomy, Turner syndrome, Down syndrome. Sex chromosomal trisomy, allosomal trisomy. Yes, absolutely. Remember, if this type of terms come in the exam, don't confuse. Autosomal trisomy, Down syndrome. Allosomal trisomy, Klinefelter syndrome. Allosomal monosomy, Turner syndrome. Remember. Clear? Next, what are the important last question? Look at here, all of you. Last question. Given below karyotype update analysis of a fetal cells analysis. Chromosome number 1, 2, 3, 4. I am getting abnormality in 21. Look at your trisomy. Look at your trisomy. So I can say, sir, the given karyotype is identified as trisomy because 21st chromosome is 3 chromosomes are there. Instead of 2 pair, 3rd chromosome also. That means 3 chromosomes are trisomy. Read the question. Based on the above karyotype, what is the chromosomal detection of unborn fetus? Down syndrome. Trisomy, Down syndrome. Write the symptoms. We discuss this one. And why? How is the chromosomal disorder different? Look at here. Answer this one. Option C. Careful. Look at here. Option C. How chromosomal mutations, chromosomal disorders are different from the point mutation or Mendelian mutation. Students, carefully listen here. In point mutation or Mendelian mutation, one base pair alter, not entire chromosome. Did I say entire chromosome? Did I say entire chromosome change, entire chromosome undergo mutation? No. In case of chromosomal mutation, one complete chromosome or chromosomal pair will be delete or add. Aneuploidy. We can write 2n-1 or 2n+1. Aneuploidy this is. Got my point? One entire chromosome will be add or entire chromosome will be delete. Got my point? But in case of point mutation, Mendelian mutation, one base pair alert. And that will be go to the next generation to next generation. It will be past the generation. But chromosomal mutations are rarely. For example, you take a Klinefelter syndrome, there is no chance of next generation formation because the sterile peoples are there. Right? Trisomy of Down syndrome, there is chance because trisomy women will produce 2 type of eggs. One egg with 23 autosomes. One egg with 22 autosomes. That is trisomy woman. So remember, that's why Mendelian mutations to chromosomal disorder difference is there. Next, what are the important diagrams you should follow in the principles of inheritance? My dear children, carefully sex determination of this diagram in fouls. Can you please tell me male heterogamity or female heterogamity this is? Male or female heterogamity this is? Female heterogamity, ZY type of what? Sex determination. This diagram also you should not ignore, and sometimes they give a child diagram and also karyotypy also. So these diagrams are thoroughly prepared from where? Principles of inheritance. We cover incomplete dominance, everything except multiple allele. I did not discuss because multiple allele, blood group crossing, everyone know. So entire chapter I touch, and important chapter I am going to start now. Stay here, carefully listen what I am discussing about the transcription, translation, lac operon, genetic code, everything. So look at the next question. Next one, molecular basis of inheritance. In molecular basis of inheritance, you know children, 3 chapters, 20 marks of weightage. Principle, molecular, evolution, 20 marks of weightage. Molecular basis of inheritance, first DNA, structure of DNA, nucleotide, nucleoside, adenine, guanine, cytosine, DNA model, completely finished first. After that, packaging of helicase in packaging of DNA helicase, nucleosome structure, finish. Diagram of nucleosome, finish. Okay, next the search of genetic material, especially in this one, Griffith experiment of transformation, plus you should also read Martian Martha Chase experiment thoroughly. Next, DNA replication, machinery of DNA in S phase, DNA replication takes place. DNA replication, enzymes, that means from which pole to which pole, from which prime to which prime it's synthesizing, which enzyme, everything you should prepare. The most complicated important topics here: DNA replication, transcription, genetic code, translation, and HGP, lac operon, fingerprinting. This all parts you should not miss, my dear. Thoroughly, line by line. In biology also, 1 or 2 questions they may give tricky. They may give higher order thinking. They might come from the molecular or biotechnology or biotechnology applications. Look at the first question. First question of principles of molecular basis of inheritance. Differentiate between template and coding strand of DNA. In the DNA, 2 strands are there. Double stranded DNA, 3rd prime and 5 prime. So I am writing this as a 3rd prime DNA. This is 3rd prime to 5 prime, 5 prime to 3rd prime DNA, right? Look at here, 3rd prime to 5 prime DNA I am calling the template strand, 5 to 3 prime I am calling the coding strand. Can you please tell me the first difference between these two? Sir, template strand undergo transcription. Template strand undergo pre-translation events. That means transcription. So template strand eventually give mRNA. 5 prime to 3 prime coding strand will not transcribe. But the important information about the protein, the codon, the information is written on where? Coding strand. Got my point? Template strand transcribe to give mRNA, it will translate to give a protein, but coding strand will not transcribe, but the exact information in the form of a coding strand. Am I clear? Till here everything clear? Now they are asking difference between the template and coding strands of DNA. Fine. Next, state the functions of the following prokaryote. This is the one very not only prokaryote here, in eukaryote also we will discuss the both RNAs. Tell me how many types of RNA? 3 types of RNA, right? What are the 3 types of RNA? Tell me. One is called mRNA, messenger RNA. One is called tRNA. One is called rRNA. 3 different types of RNAs are required for the transcription process. Done? Carefully listen, student. What they are asking here, state the functions. tRNA. Shall I call the tRNA is that RNA called adapter molecular RNA, which can read the mRNA and bring the amino acids. It can transfer the amino acids. Let me take one small question here. Here, this is mRNA. This is 5 prime and 3 prime. mRNA here it is A, U, G is there. This is codon. Codon is here. Look at that. tRNA will come. tRNA have anticodon. tRNA have anticodon. So anticodon is what? Tell me. Here it is. I can write UAC. So UAC anticodon. This particular tRNA read what is on mRNA, go bring the amino acid, give to ribosome. So ribosome will make a peptide bond and make what? Polypeptide chain. So what is the function of tRNA? tRNA function is can read the codon and bring the transfer what? Tell me. Amino acid. That's why the tRNA called adapter molecule. Smallest RNA. Next rRNA. rRNA called the ribosomal RNA present in the ribosome. The major function of rRNA, peptide bond making. Look at here all of you. Let me take here. This is larger subunit of ribosome. Fine. And this is a smaller subunit of ribosome. Right. Now here smaller subunit of ribosome mRNA come and bind. This is mRNA 5 prime and this is 3 prime. Remember all of you students, mRNA binding site is in the smaller subunit of ribosome. mRNA binding site is smaller subunit of ribosome. So look at here. For example, here it is AUG is there. Who will come? Here there is a tRNA. Look at here. tRNA will come with the proper amino acid. What is the amino acid? Methionine. Methionine, amino acid will form. Right. Now next, number 2. Here I am writing the second codon is, for example, UUU phenylalanine. Next here tRNA will come here. tRNA will bring the phenylalanine here. Look at here. tRNA will bring the phenylalanine. This is phenylalanine. This is methionine. So between these two amino acid, who will make a peptide bond? Ribosomal RNA is that RNA. You know which rRNA? NCERT, if you clearly read, 23 sRNA. Make a peptide bond between the two amino acids. So what is the function of rRNA? Making a bond between the two amino acids and form polypeptide chain. Are you with me? Are you with me? Are you noting down or not? Do not listen simply. This type of questions can repeat, student. Take a pen and paper and write down smaller subunit of ribosome to which the mRNA come and bind, but the polypeptide chain will form from the larger subunit of ribosome. Done. Come to here. What the question is telling us. State the function tRNA and rRNA functions. Come to the C. I will be wait for your answer. Can you do this calculation part of C? Come on, Allah, Sania, any one of you, can you do this calculation part? Can you do this calculation part? What is the question is given here? A DNA segment has 1500 nucleotides. Out of which 410 are guanine containing nucleotides. A is there, 410 adenine is there. And how many pyrimidine bases are given? Pyrimidine means, look at here, I can write C and T, both are there asking. They're not only asking a cytosine, not only asking a thymine, both they are asking, that means C and T. I will wait. Come on, any one of you. Come on, Kitraj. Any one of you. Any one of you. It is DNA. U is not there, Kitraj. U is not there. This is DNA. They're asking a DNA segments, right? In the DNA, U is not there. So C and T only. Very good. Who is this? Raina. Shadi, absolutely correct. Look at here what exactly. Now, according to Chargaff rule, tell me here 1500 nucleotides are there. I will give the hint, you can do. Now, out of this one, 410 are A. A equal to T, right? Adenine equal to thymine or not? A equal to, the number of adenines are equal to number of thymine. Next, so I am writing 410 is what? Tell me, here it is T. So here it is 820, right? 1500 minus 820. Come on, answer me. How many of C got? How many of you see what is C and what is T? Come on, any one of you. So we can look at here, out of this one 1500, 820, right, 780 or 780, out of the 780 what? C is 340, 340 of T. Yes or no? Look at here, 340, time is what? 410 plus 340 I think, right? 340 is C, 410 is T. Yes or no? Am I clear? Are you following me? Abin, Shadi, clear? Nandita, till here everything clear? So we can write what is the number of cytosine and thymine both. Cytosine and thymine both, if you calculate here, 740 or 700, this is 750, right? 750. Yes, 750. Done. Right, so this is they're asking what, go back to this one. Here, how many pyrimidines? C and also T. They're asking already 410 is given. 410 is A, so 410 is T. T is a thymine. T is a thymine, a pyrimidine is there. Right? So out of this one, if you subtract, automatically our answer will come. So next question. Look at here, beta. In this question, they did not given a 5 prime, 3 prime, nothing is given. But they given a small hint to us. What is the small hint? Carefully look at here. What is the small hint? Construct a complete transcription unit. Wait, all of it. Complete transcription unit with promoter, terminator, base pair, hypothetical, something is given. Template strand given. Template stand given below. That means we got a template stand. What is this template stand? That means I can write 3 prime. Tell me here A T C G. A T C G. G C. A T G C. Next A T C G. A T C G. Right? A T G C A T A C. This is 5 prime. What the code is this one is called template stand. Template stand is given. Transcription unit they are asking. So before this one, let me write here, beta. This is 5 prime coding strand. T A C G T A C G T A T G, and this is 3rd prime. Shall I write here below, before this one, promoter is there? Promoter, right? And here I am writing this is terminator. So terminator plus promoter, template and coding strand is become complete transcription unit. In NCRIT, transcription unit is given like this. How? Look at here. This is 3rd prime, 5 prime. This is 5 prime. 3rd prime, upstream, downstream, promoter, terminator. Am I clear? This is a template strand and this is coding strand is given. Am I clear? This is a transcription unit diagram is given, but they given DNA strand. So you have to make template to coding, make a double stranded DNA, promoter one side, terminator on the other side. This is a complete transcription unit. Till here everything clear? Should we memorize this 6 to? No need. All 64 codons, no need. Few codons, like for example, if I am asking here what is a stop codon, you must know what is stop codon. We will discuss that also, we will discuss in a minute. What is stop codon, what is start codon, and U is code for what? Certain few amino acids, a few codons only you should remember. Not entire salient feature of genetic code is important. All the features, all the salient feature you should remember. So what is the answer of this one? The construction of transcription unit. This is the transcription unit. I am telling you, student, from molecular base of inheritance chapter, transcription, translation topic thoroughly prepared. They can ask a different, I will discuss today different type of question. This is one of the important question. Come to the second one. Write the RNA strand from the given this one. Can you get the RNA strand? Can you give me what is the RNA strand can come from here? So I am erasing this one. Can you tell me what is the RNA strand? Sir, this one will be transcribed, right? So I can write 5 prime. Yes or no? RNA and DNA will be 5 prime to 3 prime. So tell me now here 5 prime. This is U. A, C, G, U, A, C, G, U, A. This one is U, and also this is G, 3 prime. This is called mRNA. Till here everything clear? Till here everything clear? No doubt. Till here everything mRNA stand is done or not? Clear? Shall we go to the next question? Students, come on, answer me. Kidras, till here everything clear? Look at the mRNA and the transcription unit. mRNA and the transcription unit. What is the next question? Look at the next question. What is the next question? Can any one of you do this? Can any one of you do this? How is hnRNA processed to form a mRNA? Asking. How hnRNA processed to form a mRNA? Wait a moment. Here, this is 3 prime, 5 prime DNA, 5 prime, 3 prime DNA. So this is template and coding strand here. From this DNA, we will get one strand called first hnRNA. This is hnRNA. This is not double strand, single strand only. hnRNA. Heterogeneous nuclear RNA. This heterogeneous nuclear RNA to 5 prime capping takes place. This is called capping. What is capping here? Capping here. Tell me, this capping is what? Methylated guanosine unusual nucleotide, right? Methylated guanosine unusual nucleotide. Come to here, beta. What is this one called? 3 prime. 3 prime is called tailing. 3 prime is called tailing. Tailing is A like that. What they call? Many adenines are there. So addition of capping and addition of tailing at 3 prime and 5 prime plus wait a moment. This hnRNA has, if you are able to recall, exon, intron, exon and introns are there. Come on, answer me. This hnRNA undergo a process called splicing. In this splicing, introns will be removed, all the exons will be joined to become matured mRNA. You must write this entire content with the diagrammatic manner. You have to write the content and below that one you have to mention the diagrammatic manner also. hnRNA convert into mRNA after splicing, after adding of capping and also tailing. Splicing is absent in where? There is no splicing in what particular organism. Which are there? There is no splicing. Arif, there is no splicing in particular organism. Any one of you tell me where is no splicing, no exon, intron, deleting? Prokaryote, that means bacteria. Done. Draw a neat label diagram of nucleosome. Nucleosome is made up of, we know, we have discussed this one. Nucleosome is made up of 4, that means octamer, right? 4 are in the form of 2. They are in the form of 2. These are H2A, H2B, H3, H4 are there, and the DNA will be wrapped around like this. And you can follow NCRT, and here there will be a H1 proteins or histone proteins are present. Nucleosome made up of DNA plus histone octamer protein. The DNA which wrap around the histone we call core DNA. Core DNA. This one is called H1 protein. This one is called nucleosome. Remember, all of you. Third question. Next question is what? Mention what able to histone is acquired positive charge. Before I go to this one, let me ask a small question. Can you please answer me? Histone proteins are made up of how many amino acids? What type of amino acid? Which amino acids are rich in histone protein? Tell me. Which amino acids are rich in this histone protein? Lysine plus arginine. Yes or no? There are 2 amino acids are there. What are the amino acids here? These amino acids are lysine amino acid and also arginine amino acid. This lysine and arginine amino acids are positively charged amino acids and basic amino acid. If you are able to recall biomolecule chapter, we learn acidic amino acid, basic amino acid, neutral amino acid. Can you please tell me how positively charged this nucleosome or histone protein got? Sir, because the histone protein rich in lysine and arginine, positively chargeable amino acid. So negatively chargeable DNA wrap around the positively chargeable, what, tell me, histone protein. Remember all of you, these questions till here, everything clear, no doubts, everything done. Look at the next question. This is a complicated, very important question. Look at here carefully. All of you carefully look at here what I am teaching now. Look at here in this question. Allah, identify the polarity of the diagram A to A. Polarity, this A and A they are asking. So what is the polarity? Come on, Kitraj, what is the polarity? Sania, A to A, what is the polarity of this mRNA? Can you please? Yes, very good, absolutely. So here I can write, wait a moment, this one. For our understanding, I am doing what? This one is a smaller subunit of ribosome. This one is smaller subunit of ribosome. Smaller subunit. Ribosomal unit, right? Ribosome, fine. What about this one? Larger one. Larger subunit, done, fine. So I can write, we know every time, remember, the left side one is the 5' of mRNA. 5', 3'. Just now I told you the small mRNA attached to the smaller subunit. That means the binding site of mRNA in smaller subunit. Done. Now this is, I am writing 5' and this is 3'. Done. First question answer is done, right? They mentioned what? Identify the polarity of A to A. Fine. Very good. Next, after this one, what? How many more amino acid expected to be added? Think carefully, think and answer me. How many more expected, Shadi? Read the question carefully. Read the question. How many more amino acids, Sania? How many more amino acid? 0. Albert, 0. Kitra, 0. Other students, any one of you? Sania, careful, look at here. Sir, here, how many more amino acids means what? Here we have a codon. Last codon is what? Last codon is UAA is given. Look at here. UAA is given. Look at here. UAA. Can you please tell me what is UAA? UAA code for what? UAA code for what? Come on. Stop codon. It is a stop codon, right? So UAA, I am writing it is a stop codon so that this one is no more amino acid added. Beta, carefully listen, carefully look at the diagram. Look at that codons also. Before start codon, what is the codon is there? Before start codon, AGU is there. Look at AGU. AGU code for serine already given. AGU code for serine already given. So last amino acid is serine only. After that, no. Because there is a UAA is there. So no more amino acid added. Got my point? Come to here. Next one. Mention the DNA sequence coding for serine and the anticodon of tRNA of the same amino acid. Coding for serine is what? Already given. Serine coding is what? AGU. AGU. What is the DNA sequence then? I will check. Chalo, how many of you, if you give the proper answer, that you understand this question thoroughly, what is the DNA sequence? Anyone? Very good, absolutely very good. What about others? Allah, come on, Sania, Kitraj. The answer is TCA. Look at this one is, I am going here. What? This one is paired with T and this one is C and A. So the DNA sequence is TCA. DNA sequence TCA here, and anticodon, how can I write the anticodon now here? Sir, AGU anticodon is UCA. A is the anticodon. Anticodon present in where? tRNA. Are you with me? Last question. Why are some untranslated sequence bases seen in the mRNA? These untranslated regions can increase the efficiency of translation. Untranslated regions are present before start codon and after stop codon, increase the efficiency of translation. Remember. Till here everything clear? Done. Next question. Look at the next question. Answer the following question based on the Michelson and Stahl's experiment. Michelson and Stahl experiment they are asking. Semi-conservative experiment. Michelson-Stahl semi-conservative experiment in the what they call Escherichia coli bacteria. First, write the name of the chemical substance used as source of nitrogen experiment by the what they call by them. So what is the chemical? Shall I absolutely, as in very good. We can write what? NH4Cl, ammonium chloride. First question done. Second one, what tell me? Why did the scientist synthesize the light and heavy DNA molecules in the organism used to be experiment? To understand semi-conservative, why light and heavy can be easily identified by the density gradient, so that half of the DNA is a parental DNA, half of the DNA new DNA. That's why we call the semi-conservative. Done. Next, why did the scientist make it possible to distinguish the heavy DNA molecule from the light DNA molecule? Heavy DNA, why did the scientists make the possible to distinguish? Because this distinction can clearly tell that newly synthesized whatever after replication of DNA, that is not entirely new DNA, that is not old DNA. So what is that? Partially, that means 50% old DNA, 50% new DNA. That's why we call semi-conservative. If it is a completely new, we would have called the modern DNA. If it is completely old, we would have called that what they call conservative DNA. But why we are calling it is a semi-conservative DNA? Because this semi-conservative 50% parent, 50% newly synthesizing DNA. Next, write the conclusion that scientists arrived at after the completion of experiment that DNA replication is semi-conservative enough. What they concluded at the end of this experiment, they collected the sample every after generation the NCRT is given. After a few generations, they collected the sample and they identified with the use of cesium chloride the density gradient. They got the every generation is what tell me what they call semi-conservative DNA. So they concluded the Michelson-Stahl experiments finally concluded DNA replication is semi-conservative. Experiment questions can also repeat. Remember. Next question. Oh my God, lac operon, prepare different sample paper, different type of questions from the lac operon. Lac operon discovered by Jacob and Monod. This is a gene regulation in the prokaryotic lactose operon. Carefully look at this question, what they given. Students, study the schematic representation of a genes involved in the operon, lac operon given below. P, promoter, regulatory gene. I, repressor, promoter. Next, what they call operator, LAGZ gene. Next, permease enzyme gene, transestylase enzyme gene. Identify and name the regulatory gene, this operon. Explain its role in switching of the operon. Which gene is the regulatory gene? Sir, I gene is the regulatory gene. I gene, I can call this I gene is the gene we called regulatory gene. This regulatory gene is that gene switching off when inducer is not there. When inducer not there, can you please answer me, beta? What happened? Explain, name the regulatory operon and explain the role of the switching off this one. Now, how it will switching off this one? This I gene undergo transcription to form mRNA, translation to form repressor protein. This repressor protein go and bind to where? Operator, and switch off the operon. If it is this repressor go and bind to operator, RNA polymerase enzyme cannot slide over the structural genes, over the ZYA gene. So no mRNA, no beta galactosidase enzyme. So the operon is off mode. Question number 2. Why is operon regulation refers to as a negative regulation? Operon is negative regulation because the repressor protein go and bind to the operator to turn off. The repressor go and bind to the operator to off. So it's a negative effect, negative operon. Next, name the inducer molecule and the producer of Z gene. So name the inducer molecule and product of the gene Z. Can you please at least this answer me? Inducer is what? What is inducer here? Sir, the inducer of in the lac operon is simple allolactose or lactose. Lactose or allolactose. Very good. So I'm writing here what tell me? The producer gene is Z is beta galactosidase, right? Beta galactosidase. And Y is permease of the operon. Write the functions of these gene product. Y gene permease enzyme allow the lactose from outside to inside. That means it increases the permeability of plasma membrane toward the lactose. Z gene, beta galactosidase enzyme. Beta galactosidase enzyme is essential for the digesting of lactose into glucose and galactose. Enough. Not only this question, I selected this is a simple question based on lac operon. Different type of questions can come. Student Ala, Shadi, Raina, I am requesting you all prepare this question, this lac operon thoroughly. Azim, I hope you all are following me. Next question. Look at the next question. State the functional difference between the following codons: AUG, UAA, specific and degenerated. Specific degenerated is belongs to where? This silent feature of genetic code, right? Now let me take this one, AUG. Sir, AUG is a start codon and it is also code for the methionine. So dual function. UAA fellow, who is this UAA? Stop codon. This is called start codon. Done. Specific and degenerate. What is the meaning of specific and degenerate? Genetic code is specific. That means one codon is specific for one amino acid only, but one amino acid code for more than one codon. Let me take one example. UUU code for phenylalanine only. Next to UUC also phenylalanine. That means one amino acid have more than one codons, but one codon is specific to one amino acid only. UUU code for phenylalanine. UUU also code for the methionine and some other. No, it's not like that. UUU means phenylalanine only. Understand? Next, it is universal also, remember. Next, differentiate between the Turner syndrome and Down syndrome. We discussed just now only. Down syndrome is the trisomy and the Turner syndrome is monosomy. It is allosomal, it is autosomal. One chromosome will be less here, one chromosome will be more here. Here, whatever the effects are there, the effects you can write the characteristic feature. The Turner syndrome in the females, you can write the characteristic feature here. Come to the last one. Describe the termination process in transcription of bacteria. Wait. All of you look at here. You can get the question of initiation part, elongation part or termination part. They are asking in the bacteria. So row factor you should remember. Look at here. This is what they call transcription unit I'm talking about. Here this is a promoter. Exactly I'm writing here. This is a 3' and 5' template stand. Exactly, here there will be a sigma factor and RNA polymerase enzyme attached. When the new RNA synthesize, mRNA synthesize, exactly at the terminator area, there will be a factor we call what? Here I am writing the factor called rho factor. After elongation process of mRNA, RNA polymerase rho factor will stop or terminate the elongation of mRNA. So RNA polymerase enzyme and rho factor, they will stop this what? Synthesis of a new RNA. That means here promoter, look at it, this is a promoter. At the promoter area, it will initiate sigma factor, RNA polymerase enzyme. It will new nucleotide come and start adding. So it will form a new RNA. Look at here, new RNA will form 5 prime to 3 prime, new RNA. At a particular time, this is called a terminator area. Whenever terminator area come, rho factor will stop the elimination, stop the elongation. So we can say termination of transcription. Who will terminate? Rho factor terminate. Remember, initiation, elongation, termination. Done. Next question. This is a repeated one. Now look at here, this one. Lac operon, second type of question. Look at the second phase of lac operon. First type of question we discussed. Look at the second type of question. What is the second type of question here? What answer? Okay, this is satellite DNA they're asking, and study the figure. This second one we will do later. Look at what are satellite in a genome. Explain their role in DNA fingerprinting. My dear students, I told you the beginning of this session, DNA fingerprinting mechanism is one of the important topic in molecular base of inheritance. According to Human Genome Project, 99.9% of DNA which is similar in all human beings, we called bulk DNA. We have a bulk DNA. There is 0.1% of DNA which is repeating again and again. Large amount it will be repeat in our genome, but it not code for any protein. Read NCERT carefully what is given. Satellite DNA is that DNA which is not code for any protein, and a large portion is present in the DNA. And number 3, satellite DNA is the particular sequence will repeat again and again. In my body, one particular sequence will be repeated particular number of times. In Kirtraj, in Allah, in Sanya, different times it repeat. Based on the satellite DNA, we can identify the individual. We will inherit that satellite DNA from our parents, but it is not code for any protein, remember. One more important question also I will cover. Look at here. This satellite DNA is classified into mini DNA, mini satellite, and also microsatellite. Microsatellite DNA, mini satellite DNA based on 3 aspects, 3 major what they call criteria. You will get this question, right? Based on what criteria satellite DNA is divided into micro and mini. Student, remember this satellite DNA, here I am writing this satellite DNA is that DNA, number 1, AGCT ratio. What is the ratio of AGCT? The second point is how many times, number of times, how many times it's repeating. Number 3, length. Let me take one small example. ACGGTA. One small sequence. One more time repeating. One more time repeating. One more time. So like that 10 times repeating. So the length will form. Yes or no? For example, 20 times repeating, length is big. So based on 3 criteria, remember students. One is AGCT ratio, length, and also repeating times. Come to B all of you. I don't want to waste time. Look at here, all of you come to B. Study the given figure and answer the following question. So mRNA, what is this X? And what is this? What they call this? All mRNAs are given below. Okay, look at here, study the given figure below, answer the following question. Lac operon is given, that is we clearly understood this lac operon, XYZ, beta galactose and everything. Look at the first question. Name that molecule X. Sir, X is inducer. Yes, sorry, X is repressor, right? X is repressor. I'm sorry. This one we can write what? This is repressor protein. Synthesized by I gene. I gene is giving repressor protein, inhibitory protein. Are you with me? Are you there? I is repressor, right? Good. Now, how does this molecule get inactivated? Can you please tell me? How does this inactivated? Yes, come on. How does it inactivated? That means the I gene inactivated when inducer present, right? When lactose is there, I gene repressor protein will be inhibit. I gene inhibit, this protein will be what they call stop. Look at it. How does it inactivate when lactose is there? Right, next one. Which one of these structure genes code for beta? Oh my god, very simple. Lac Z gene. How this type of questions come, students? I hope you will get full out of full marks, so we can rock with our final examination result. Especially in bio, you can score more marks. Now listen, before you go, every topic thoroughly prepare a question they ask. What question, what gene, what codon they are asking, which gene they are asking, we should be answer. Okay. Now, when will this transcription is the gene is stop? When will the transcription of this gene stop? Sir, when will this transcription will stop? When lactose is there. When lactose present, I gene will stop the synthesis of what they call transcription. Yes or no? When I gene will stop, when lactose is not there, I gene undergo will be the repressor protein will form and go to a operator. When lactose is there, I gene no need to what? Synthesis repressor protein. And one more time telling you, lac operon questions, transcription question, translation question, thoroughly prepare. Next question. Look at here. Next question, genetic code. Somebody asking, sir, all the codon should be? No. Look at here. I'll tell you the hint. Here in this one, first UAA, UGA, UAG, stop codons. You should remember, right? Stop codons. Look at these three stop codons. Like one more codon, AUG. Okay, next AUU, right? Next one is UGG. These two codons are single codons for single amino acids. Look at here. If you take this diagram carefully, look at here. Tryptophan, UGG, only one is there. Next AUG, also only one is there. In case sometimes if you get a question, in the entire 64 codons in the genetic code, which all the amino acids are code for single codon? Methionine, tryptophan. Okay. Now, how many codons code for amino acid? 61. How many do not? 3. Total how many are there? 64. Explain the following, given one example of each. Explain the following, giving one example of each. Now here it is. Unambiguous and also specific codon. Unambiguous specific codon. One codon exclusively code for one amino acid only, but one amino acid has a many codon, more than one codon. More than one codon. Unambiguous. Specific means we can say the specific codon. For example, I'm talking about UGG is code for the tryptophan only. UGG also code for valine. No, it's not like that. No, not like that. Example, you can write any one of codon. Degeneration codon, I told you just now, degeneration codon, one codon code for one specific amino acid, but that amino acid have many codons. Next, universal codon. Universal codon. Codon is universal. That means, careful listen here. Careful look at here. In my mRNA, UUC, GGG is there. Just assume how many 3 codons are there? 3 amino acids. Phenylalanine, and GGG, I think so it's glycine. I think glycine, right? Glycine. right? Same mRNA if in the bacteria, same mRNA in the other animals, also same amino acid. Because of this reason, if you able to recall biotechnology application chapter, we can produce a human insulin in the bacteria E. coli. It is possible because the genetic code is universal. Genetic code is what? Universal. So we can write universal. Initiation codon is simple AUG. I can write initiation codon. Next question. Now look at here, this type of question, molecular base of inheritance, this type of smart question they will give. Carefully look at here. Illustrate the given DNA segment which constitute a gene. This is a gene and this one is a dark shading and white shading is given. Name the shaded and unshaded region of the gene. What is this shaded and unshaded? We can write shaded intron, unshaded is what? I can write exon. Exon, intron, so I can write HNRNA. Heterogeneous RNA, exon, introns are there. Done. Now explain how these genes are expressed. How these genes are expressed when this HNRNA undergoes splicing, the non-coding part intron eliminate, the exon point join, they make a specific protein. That's how they express their character. How is this gene different from the prokaryotic? Now, any one of you answer this one better. Third one, how is this is different from the prokaryotic one? Simple. Sir, in the prokaryotic transcription, no intron, no exon, no coding, no coding and no non-coding parts are there. But this is a eukaryotic transcription. That's why intron, exons are present. Are you following me? Allah, you got my point? Yes, absolutely correct. Absolutely, Albert, clear, Keetraj, everything clear? Now, remember this type of question can ask. Don't get confused. Look at the question carefully, read and finally answer. Next one. Very last question from the molecular. The RNA polymerase holoenzyme transcribe. RNA polymerase enzyme transcribe. Holoenzyme transcribe HNRNA, or I can write mRNA. So look at the four options are given. It is an MCQ question. MCQ question. RNA polymerase enzyme, the promoter structural gene and the terminator gene. No, not all this. Now B, the promoter and terminator gene. No, it is only transcribe structural gene. So structural gene it will transcribe to form what? A specific protein. So answer I can go option D. Look at the last one. If the base sequence of codon is mRNA 5' AUG, what the sequence of tRNA? Oh my God, I am shouting. Can any one of you answer what is the answer of this three? What is the anticodon of AUG? Proper correct answer of the following. I will cross check now. Any one of you? These four options are given. What is the anticodon of this AUG? Anyone? Ketraj, Ala, Sania, Shadi, Raina, any one of you? Sir, I can write tRNA. tRNA is what is this? AUG. Okay, tRNA I can write here it is UAC. So 5', 3'. So 5', 3', option A. Am I clear? Till here everything done? Yes, absolutely correct. Next question. Now, name a few enzymes involved in the DNA replication other than the DNA polymerase and ligase. Name enzymes which are participate in the DNA replication apart from these two. Sir, I can write helicase enzyme, topoisomerase enzyme, phosphorylase enzyme. Am I clear? I can also write what? Tell me. DNA polymerase 1 enzyme. These all are helper enzymes. Right? Name the functions of this enzyme. Helicase enzyme can unwind this 2 DNA strand, break the hydrogen bond and open the replication fork. Yes, replication fork. Topoisomerase can remove the tension of this strand. I'm clear? Primase enzyme essential for the primer synthesis. You can write this one. Carefully read this replication. What purpose? How many enzymes are involved in the replication of DNA? Next, there is only one possible sequence of. Look at this. Hold on, all of you. There is only one possible sequence of amino acids when deduced from a given nucleotide, okay, but multiple nucleotide sequence can be deduced from the single amino acid sequence. Explain this phenomena. What they are telling? What is this multiple amino acid? Look at here. Careful, look at here what I am telling. There is only one possible sequence of amino acid. I am writing here, this is mRNA 5 prime, U, okay. Now this is 3 prime, fine. Now I am writing here one more codon. I am writing this is UUC. I am writing this is 3 prime. Okay. Now answer me, amino acid is one only, but nucleotides change. Look at there, UUC, nucleotides change. Yes or no? If you go back to a previous genetic code one, last 4 one, look at here. If you go back to the genetic code diagram, GGU, GGC, GGA, GGG, all are nucleotides are changed, but amino acid same only, right? Amino acid glycine is same only. So what the question is telling us? The question is telling us there is only one possible sequence of amino acid. Yes, when deduced from the given nucleotides, but multiple nucleotides. Yes, multiple nucleotides are there because genetic code is clearly given. One amino acid contain many more than one codon. Codon made up of 3 nitrogen bases. If codon change, nitrogen bases also change. Nitrogen bases change means codon change. But there are chance of a same amino acid. You see same amino acid. Nitrogen bases change, codon change, amino acid is same. Are you with me? Got my point? Next question. So these are important question. Explain the process of amino acylation tRNA. First step of transcription process, student. It's a translation process, student. Amino acylation of tRNA. When amino acid is here, this is amino acid. This amino acid will be activated with what? ATP and amino acyl synthetase enzyme. So this will be taken by what? 3 prime of what? tRNA. Look at here, tRNA. It will add and it bring the proper amino acid to a proper place for what? Protein synthesis process. Amino acid, tRNA, entire mechanism you can prepare and write. Next, here now next, how do ribosomes in cells act as a factories for protein? Ribosomes are containing rRNA. Ribosomes are containing rRNA which help in the peptide bond formation between the amino acid. Between amino acid peptide bond formation. So ribosomal RNA also behave like a catalyst. Ribozyme enzyme is there. This ribozyme enzyme 23S RNA. 23S RNA ribosome can make a peptide bond between the 2 amino acids. That means ribosomes play essential role in the making of polypeptide. Making of what? Polypeptide. Remember. Next, describe initiation and termination phases of protein synthesis. Who will initiate? AUG initiate and termination. There is a stop codon will be terminate. AUG start codon will start this initiation of translation. Remember, whenever the translation comes, student, larger subunit and smaller subunits are apart, separate. Magnesium ions are helps to join these two to form dimer. So this dimer, mRNA come and bind and it started to initiate the complete translation process. Done? No doubt. Next question. Look at here. Look at the last question of molecular base of inheritance. Last question. What is the last question is given here? Look at here. Study the flow chart given and answer the following question. Can any one of you do this one? heat kill S strain plus live R strain into mice. What is the fate of the mice? Any one of you tell me. A. Allah. What is the answer of this A here, Albert? Here, heat kill S plus live R. Heat kill S plus live R. So I can write, look at here, heat kill, okay, here it is, heat kill S plus live R. Live R. So something is transferred from S to R so that it's died. It's dead, right? Yes or no? Dead, right? Absolutely. Very good. Correct. Now, heat kill S DNase plus live R. Now, heat kill S plus DNase live R. DNase enzyme adding means DNA will digest. DNA will digest. So answer me. Tell me now here. Now into a mice will be survive, right? Mice will be survive or mice will die in B? In B, mice will survive or mice will die. Answer me. Come on, student. Albert. Come on, Ayan. Now answer me. Sir, it is that this one is enough. Yes. This type of whatever the question I discuss here, these are from all the topic of molecular basis. You can also practice more questions. This in this 2 hours I cannot cover. Okay, now tell me, it will be survive, like a life, it can be live. Now answer this question. This type of, why I am selecting this question, this type of question, higher order thinking question can come. That's why I am discussing this question. I did not left any one topic of transcription, molecular basis student. This is for you student, for you people. Okay, when I am reading the molecular, did I cover this topic? Did I cover this topic or not? So all the topics you should cover. Now look at here, first question. What is the first question is given? Give the name of the organism and differentiate between these two R and S. Sir, R and S differentiation. Streptococcus pneumoniae bacteria, which is pathogenic in case of S. That means this is bacteria. On the surface of bacteria there is a proteinaceous coat is there. This protein coat is pathogenic. Remember, that's why this is a S phase. What about the R? R is a what they call rough variety. No protein coat is there, so non-virulent or non-pathogenic. Write the result of AB. Write the result of AB obtained in steps C, D. Where is C? This is C, this is D. Here it is live, here it is dead, here it is live. Am I clear? Next, give the name of scientist who performed the step ABC. Who performed the steps ABC? Chalo, I am giving to you all. Can any one of you? Any one of you tell me A, B, C. This is A. This is A. Okay. And this is B. And this is C. A, B, C. Come on. A, B, C scientist. Griffith. Yes or no? Griffith. Yes, absolutely Griffith. Then who have done the D? D is done by his what they call 3 scientists: MacLeod, McCarthy, Avery. MacLeod, McCarthy, Avery. So the answer is what? Griffith. Write the specific conclusion drawn from the step D. Where is step D? This is, sir, the conclusion from step D. DNA is the genetic material which is transferring from S killed bacteria to R, so the R become pathogenic. Yes, killed one, DNA is transferred to R, it will become pathogenic. The conclusion, my dear student, DNA fingerprinting only I not explaining here. The rest of all parts I covered. DNA fingerprinting, read by yourself, and the what they call case studies, the reports of DNA bands and everything done. So this is 2 chapter, principles of inheritance, molecular base of inheritance. Now molecular base of inheritance, these are the diagram you should cover. transcription unit of diagram, replication diagram. This is called Hershey-Martha-Chase diagram, and also nucleosome diagram. These diagrams carefully prepare and go. And 10 minutes hold a simple explanation of evolution also I will give. Do not leave, okay? Only 10 minutes. Done? Clear? Next, look at here. This is a diagram. This is a genetic code. Especially this diagram, student. This diagram. Everyone, please, all of you. Translation diagram. Carefully prepare and go. Where exactly amino acids are adding. Codons are there. Small unit, larger unit, polarity. Look at this polarity. Carefully prepare and go. Next. Lac operon also, very important. Lac operon also prepare and go. In case of evolution, look at here. Please do not leave the group, do not leave this session. Listen here. After this evolution you can do, because this chapter 20 marks weightage. This is very easy. Evolution very easy. Very small questions I selected. Evolution, origin of life, biotic theory, Operon and Hadley theory, what they call organic evolution, and biological evolution. What are the evidence of evolution? Embryological evidence, anatomical evidence, biochemical evidences. Got this all evidences you have to prepare properly. Adapt, especially this adaptive radiation, Darwin explanation. Adaptive radiation pinches. Right? Next, here Hardy-Weinberg equilibrium, P square plus Q square plus 2PQ equal to 1. Very important one. Next, origin of human, man evolution. This all the topics cover, especially adaptive radiation, marsupial adaptive radiation, and placental adaptive radiation. Darwin pinches, natural selection. Properly prepare. Look at the first question. Urey-Miller experiment is this. This is Urey-Miller experiment is proving that organic evolution, the life origin in water, and by the fusion of your chemicals are there, they are adding the chemicals. What is exactly his experiment? Look at here. Can we call human evolution as adaptive radiation? No, students, no. Adaptive radiation is that process, the origin of a different species geographically from a common ancestor. If you take this is a common ancestor, from this common ancestor the species are radiating. Species number 1, 2, 3, 4, 5, like this. And the geographically they migrated and they become a reproductively isolated, they become new species. But in case of human being, it's not like that. So we are not considering human evolution is adaptive radiation. No, it is not adaptive radiation. Study the diagrammatic representation and Urey-Miller experiment. Okay, this is Urey-Miller experiment you give it. Look at the first one. What is given? How did Miller create conditions which existed before origin of life on the earth? He created temperature in a spark chamber, and in that spark chamber lightning, that means light, with this one, look at here, it is given what they call beta energy, that means electrode they added, that means temperature he created. Chemicals also he added. What are the chemicals he added? Hydrogen, ammonia, and also what they call helium, hydrogen, ammonia, and also water vapor. Water vapor, hydrogen, ammonia, ammonium he added. Am I clear? So 800 degrees Celsius of temperature he created, he got that amino acid as a result. So look at what they're asking. Name the organic compound formed in the collection of end of his experiment. 18 days after his experiment he got chemical compounds like a glycine and aspartic acid, like a amino acids. Amino acids he got. Mention the kind of evolution is experiment support. Organic evolution, which was given by Operon and Hadley. Got my point? Urey-Miller diagram, thoroughly prepare. Urey-Miller experiment also read and go, students. And here, what the ratio? Here it is given. Water given. Ammonia is given here, right? Ammonia is given here. I think it's what they call hydrogen also given here. Water vapor also given here. And methane also given, I think. Methane also he added. Next one. Select the homologous structure from the combination given below. Homologous structure combination given below is given. Homologous structure. Now, forelimbs, whale, bats, tuber of potato, sweet potato, eyes of octopus and mammal, thorns of bougainvillea and tendrils, which is called homologous structure. There are homologous structure. We know homologous structures are that structures which are structurally similar but performing function is separate. So what is the answer I can write? Yes, very good. Tell me what is the answer I can write? Forelimbs and whales, bats, tuber and potato, eyes of octopus and tuber of potato. These two are function is same. Shall I call the analogous? Analogous. So here I can write what? Tell me. Forelimbs of whale and bat 1 and 4 I can write homologous. My dear children, evolution chapter, especially homologous, analogous, different type of example prepare. Homo, ana, different type of example prepare. And homologous organs are the examples of what kind of evolution? Divergent evolution. Look at here. State the kind of evolution. Divergent evolution. It is diverting. Divergent evolution. Analogous organs are convergent evolution. Done. Next question. Look at the next one. Can you answer this question? The proper pathway of the evolution of human being. The most accepted line descendant in the human evolution. Australopithecus? No. Actually, there are 7 steps. Are there 7 evolutionary stages? What are they? Ramapithecus, Dryopithecus, Ramapithecus, Australopithecus, Homo habilis, Homo erectus, Neanderthal man, Homo sapiens, right? So based on this one, any one of you, right? Very good, very good. So we can write, first one we can write what? Tell me here. Ramapithecus, Homo habilis, Homo erectus, Homo sapiens. So answer C. In this one, carefully look at here. Ramapithecus is more man-like 15 million year back. 15 million year ago. Homo habilis, Homo erectus. Homo habilis 650 cc cranial capacity. Homo erectus 900 cc. Remember. Homo habilis, pure vegetarian. Homo erectus started to eat meat 1.2 or 1.5 million years ago. Okay, next. In a certain this question, everyone carefully listen here. This question, do not leave. Carefully look at here. Hardy win by equilibrium question. This is genetic population genetics. What is the ratio of allelic, the frequency they're asking? Carefully look at here. In a certain population, Ketraj, Sania, everyone carefully listen here. What exactly the Hardy win by equilibrium? Allah, Albert, everyone look at here. In certain population, the frequency of 3 genotype is, what is that? Capital B is 22%. Capital B, small b is 62%. Small b is 16% is given. In the population, 3 different type of gene ratio, the frequency given. Listen, what is the likely of frequency of capital B and small b allele? They are asking you to write the frequency of capital B and small b. So here capital B, here it is, I am writing capital B here, what percentage? 22. Capital B small b, in the capital B small b, what is that? 62%. Half of them are capital B is there, right? heterozygous condition, half of them are capital B is there. So half of them are capital B means I am writing it is capital B is 31. Here it is 31. So I am writing here what is how much it is, what tell me, 3, and it is by 53% of capital B allele. Capital B allele. What is small b allele frequency? Here half of the small b is there, here 16. So small b is 16% is there. Capital B small b, half of the small b also there. So I can write here it is 31. So 47% of small b. 47% of small b and 53% of capital B allele. Remember, sometimes you will get like this. Only you will get homozygous, they will ask heterozygous, and also what they call recessive they will ask. That time you have to use p square q square 2pq equal to 1 formula to identify heterozygous or homozygous allele. Next question. Last question. Look at the last question. Very simple question. State and explain any 3 factors affecting the allelic frequency. My dear children, remember all of you, Hardy-Weinberg equilibrium, p square q square 2pq equal to 1 can affected by natural selection, gene migration, recombination, mutation, genetic drift. Am I clear? These all the factors affecting the Hardy-Weinberg equilibrium. According to Hardy-Weinberg equilibrium, p square q square plus 2pq equal to 1. That means frequency of allele remain constant generation after generation. Generation after generation until and unless that population big, no migration, no immigration, no mutation, no natural selection, and random mating should take place. But there are factors which affecting this one. So you should write natural selection affecting, recombination affecting, mutation affecting. These all are affecting the Hardy-Weinberg equilibrium. Why do you understand? Sir, what do you understand from the equation p square 2pq equal to? Sir, from this equation, in a given population, allelic frequency, it could maybe what they call homozygous, capital A, small a will become 1. Heterozygous allelic frequency, homozygous allelic frequency, homozygous recessive allelic frequency become 1 generation after generation. So he represented in the form of p square 2pq q square equal to 1. Now write your answer. Observation on the variation seen in Darwin pinches is given. What is Darwin pinches? Look at this is the Darwin pinches is given. This is the one which is every time Darwin pinches question is repeating from the evolution chapter. Darwin pinches question. Look at here. Write your observation on the variation seen in the Darwin pinches shown above. Look at Darwin pinches is given here. Basically, if you understand this Darwin pinches adaptive radiation, in Galapagos Island, seed eating 14 variety of big variety of birds were living. Seed eating pinches were there. These seed-eating pinches are migrating to the different geographical area of that Galapagos Island. So the beak is suitable for few type of bird. Beak is suitable for the cactus plant eating. Few type of beak is suitable for the insects, worm. So slowly they adapted to that environment area. They become new species. That's why radiating, adaptive radiation. Understand my point? So this seed eating pinches, from this seed eating common ancestor pinches, few pinches are evolved to form a new species like a eating cactus vegetarian. Few pinches are going to the worm eating insects, worm eating pinches. Few are going to the insect eating one. Got my point? So they are radiating, geographically they are radiating from the common ancestor, and then later they become a new species we call adaptive radiation. So what understand? How did Darwin explain the existence of different varieties of pinches on the Galapagos Island? he explained that different type of pinches are existing in the Galapagos Island. The beak is in a seed-eating pinches. Initially, the Darwin pinches are eating seed, so that the seed-eating Darwin pinches beak is vary better. In different pinches, it vary. Let me take a small example. In human being, we all are human being. We all are mammals, we all are primates, we all are same species, but few are tall, few are short, few are different. Same like that in the Darwin, the seed eating pinches beak also, few bird beak is short, few bird beak is tall, few bird beak is sharp. So this variety of beak is eventually, when they adapted to, when they migrated to particular area, they started to pluck the flesh of cactus. They're staying there only, mating there only, breeding there only, making nest there only. So they become a new species slowly. Few type of bird beak is suitable for the worm eating, so they are migrating to the worm area, so they are staying there, reproducing there. So later they become a new species. That's why Darwin says these all different type of birds are evolved from the common ancestor. The common ancestor were seed-eating finches. Remember, seed-eating finches. So till here, I only covered a little portion of evolution, but all of you, the last part, that is human evolution, properly prepare, adaptive radiation, properly prepare, Hardy-Wien by equilibrium, properly prepare, and homologous, analogous organs are thoroughly prepare. So today, the highest weightage chapter unit we discussed, students. I hope this class will help you to get top good marks in the examination. I will also wait for your final feedback of exam. All the very best. Tomorrow I am discussing human reproduction, sexual reproduction in plants, and what they call one more chapter also I will select and discuss. Do not skip tomorrow class also, and whoever not attend, please share this link to your friends to boost the concept. Am I clear? And all the very best. At least one chapter finish and sleep today. Thank you all of you for attending this class. Thank you.
Good morning.
Good morning. What's all this? Your results. Congratulations to all. I am confident you'll all achieve your goals. So young man, what are your aspirations?
I want to do robotics, ideally from the Bust Institute in the US.
And you, young lady?
Sir, I love kids and I would love to learn pediatrics. I feel Boston is the best place for that.
Sumit, what about you?
Back home to India for medicine.
Good. I'm sure you'll all achieve your aspirations, be it in India or anywhere. Keep in mind, your first challenge is the entrance, whether it's IIT, JEE, SAT, or NEET. You have to learn from the masters. Brilliant Qatar is the passport to your success.
We undertake coaching for regular and repeat batches and tuition for board examinations. Brilliant Education Centre, Doha, Qatar.